So hello everyone and welcome to the journal club from the Cost Action Ethernet. Today we have the pleasure of having with us Julie Rodor from the University of Edinburgh. Julie is a senior scientist in the group from Professor Andrew Baker, and she is with her magic helping most of the team on the non-coding RNA and coding RNA as well, like transcriptomic projects. And today she's going to present a project regarding microRNA's role in smooth muscle cell biology with a lot of cool transcriptomic data. Over to you.
Thank you. Okay, I'm going to start sharing my screen. Hopefully it's going to work again when we try. Sharing. Okay. So thanks very much for the opportunity to present our work. So we had a paper published recently in Molecular Therapy, and it's entitled Functional Screening Identifies microRNA with a Novel Function Inhibiting Vascular Smooth Muscle Cell Proliferation. And it's been a team effort. So before I start, I just want to acknowledge everyone who contributed to this work. It was a collaboration with the group of Serena Zakinia in Trieste and Mauro Giacca as well, Trieste and London. And I have to say that I've been involved, so I supervised the project and also did some of the bioinformatic analysis. But for the revision, because the people who started the project had left at the time, it was really a lot of people from the team that joined and helped finalize this project. So I'm going to start with some background. And as you probably all know, abnormal proliferation of vascular smooth muscle cell following injury to the vessel wall is a crucial factor that contributes to pathological vascular remodeling. So in this picture, it's illustrated in atherosclerosis, but actually this process is also an issue across different vascular diseases, such as pulmonary arterial hypertension and also vein graft failure. And I'm going to go to vein graft failure in more detail. So what's happening during remodeling, these smooth muscle cells that are in general in a quiescent contractile state, I'm going to put my pointer as well, change upon injury to a synthetic proliferative migratory dedifferentiated phenotype. And this can be linked to different processes, inflammation, but also growth factor. So this is important in different diseases, and our group is particularly interested in vein graft failure. So it's an interesting disease with a lot of unmet needs, but also a possibility for intervention. So what is it exactly? So coronary artery bypass graft is used to treat coronary artery disease. So when there's a severe blockage, what is done is a bypass, and the bypass is done in general using the saphenous vein. And the reason the saphenous vein is used is because it's easily accessible, and in case of multiple bypass, the length is, it's long enough to do multiple bypass at the same time. So even if we know it's not the best conduit because it needs to adapt to the artery circulation, this is still really using more than 90% of the surgery. So this is illustrated here. You have the blockage. The saphenous vein is taken from the leg and used for the bypass. So there are quite a lot of surgery performed annually, so around 400,000 in US and around 12 to 14,000 in the UK. But the issue is that there is a high failure rate. So we know that within the first year, 10 to 25% of the graft will fail. It means it will be occluded again. And by 5 to 10 years, actually, it's 50% of the graft that has failed. And what's happening when there is failure of this graft? Not much, actually. So there is possibility of vascular stents, also repeat bypass grafting, but the outcome is really low and in general it's not performed. So for the patient, it consists of monitoring and pharmacological management. So at the moment, we don't have any treatment and any way to follow up with this failure. But what's interesting in this case is we have a really important, interesting therapeutic window. So when the vein is taken from the leg, it sits in the dish for 30 minutes. So we have the opportunity to treat this vein before doing the bypass. And our reasoning would be to, as we know, vascular smooth muscle dysfunction is important. And in the case of vein graft failure, there is first endothelial cell activation, and we know that it contributes a lot to the early failure. But later on, these smooth muscle cells go from a quiescent to a proliferative stage, and it will lead to atherosclerosis and late vein graft failure. So our hypothesis is that with this step here, the smooth muscle cell dysfunction, it will prevent late vein graft failure. But the way we want to treat it is directly in the theater by doing a treatment to the vein that will be implanted for graft. And we're hoping that creating this early switch will prevent late vein graft failure. So of course, for therapy, there are different opportunity and that has been developed, but we decided to look at miRNA. So just a brief introduction about miRNA in case you don't know how they work. So miRNA are small RNA, around 22 nucleotide long. They're obtained through, so they're processed, they're transcribed in the nucleus in a long transcript. There are different processing steps happening, and then they end up in the cytoplasm as a short RNA. In the cytoplasm, they bind to a complex called RISC, and the way they work is they bind to target messenger RNA. So there will be some complementarity between the miRNA and the target, and this binding will lead or to the translational repression or and the messenger RNA degradation. And the reason it goes to the repression or degradation depends on the level of complementarity between the miRNA and its target. I think the interesting part is that miRNA can regulate multiple genes. So the miRNA can bind to different genes and leads to their down regulation. So it means with one miRNA, we have the potential to target different genes, different pathway. So for this reason, a lot of people have a look at this miRNA for therapy. So actually, there's a lot of development into miRNA-based therapy. So in general, it corresponds to the modulation of the miRNA. So we can downregulate the miRNA using anti-mir, or we can also overexpress or ectopically express the miRNA using mimic. And just to illustrate, there are several miRNA that have been taken to clinical trial, to phase 1 and also to phase 2. So it's really promising. Of course, there's still some challenges. So we need to deliver in short time. So in our case, we want to deliver them in this 30 minutes time window. miRNA might have some off-target effects because they can't bind to multiple targets. So it will be miRNA specific. So it's something we need to keep in mind. And of course, if we want them to be efficient for a certain time, we need to have them to be stable. So there chemistry that has been developed for that. And of course, the mode of delivery is also important. So our idea was to develop a miRNA-based therapy to block vascular smooth muscle proliferation. And the idea would be interesting to treat cardiovascular disease. It's a big general aim. So of course, we started with a smaller ideas first to identify which miRNA mimic block vascular smooth muscle proliferation. And then we focus on vein graft failure because of this therapeutic opportunity and study them in human saphenous vein smooth muscle as well as in tissue. So we really wanted to have the best candidate. So for this reason, we decided to do a screen. So we took pulmonary arterial smooth muscle. It's because when we started the project, we didn't really have a strong idea about, we didn't decide about which disease we wanted to treat, but then we move on to vein graft failure because it was an easy option. But in principle, we say, let's test any smooth muscle and see which miRNA change proliferation. So we started with a library of 2042, which corresponded to all the miRNA that were annotated at the time of this mimic library. We transect them, we use EDU to assess proliferation, and then to assess if the cells proliferate or not, we use high content fluorescence microscopy. So at the bottom here, you can see two images just to illustrate how the screen works. So we use miRNA control, and you can see you have the cells in blue for viability, cell count, and the one in green are the ones that are EDU positive, they're proliferating. And you can see when we have our one of our candidates, now the proliferation has been reduced strongly. So based on this readout, viability and proliferation, we look at all the mimic, and this is what is shown in this graph. So first, we didn't want microRNA that reduced cell count a lot because it would mean they were toxic. So we apply a threshold for viability, and we remove 22 toxic microRNA. Then we look at proliferation, what was the change in the percentage of EDU cells. And we find 417 pro-proliferative mimics, and we had 715 anti-proliferative. And what was interesting is we found in these least microRNA that were previously described in the literature, confirming that our screen results were robust. And we decided to look at our top candidate. So we had 10 microRNA mimic that ended up with 0 EDU positive cells, so our strongest candidate. One of them was miR34A, was quite interesting, but in addition, we had several completely novel microRNA in the context of cardiovascular. So we took them forward and looked at them in different assays. So first, we wanted to add more replicate in pulmonary artery smooth muscle cell. It's the graph on the left. So exactly the same, similar setting as the screen, but only now taking our 7 candidates forward. We also look at different type of smooth muscle cell, coronary artery and umbilical vein smooth muscle cell. And overall, all the microRNA mimic show a strong reduction, no matter which smooth muscle cell we're looking at. So it means that they have the potential to be therapy or interesting candidate across different diseases. And it's where we thought a bit better and thought, okay, let's see which disease and which condition we want to focus on. And of course, vein graft failure was something that came to our mind because of this therapeutic opportunity. So we decided to switch to human saphenous vein smooth muscle cell and characterize the microRNA effect in detail. So we did the same experiment as before, took our mimic, transfected them. And to induce proliferation, we use a combination of IL-1 alpha and PGF-beta. It's something we've been described in this cell type to induce strongly proliferation. So we look at proliferation, migration, and we also look for toxicity. Of course, you don't want this microRNA to have any toxic effect to the cell. So in terms of proliferation, instead of using imaging, we still use EDU, but this time we look at flow cytometry. So with the arrow, you can see the peak that corresponds to the EDU positive cell. You can see the peak is clearly here for the miR control, but there is a strong reduction with all the 7 microRNA candidates. And this is just the quantification on the right. So this is what we're submitting for the first, in the first submission. And one of the reviewers asked us to confirm this effect using a different method, using staining. And this is something we did with MKR67. Just one example, miR control, we clearly see some signal for MKR67. But when we transfect the cell with miR323A, 3P, there is a strong decrease of MKR67. I didn't put the graph here, but it was also quantified and was statistically significant for all 7 microRNA mimics. So we confirmed there was an effect on proliferation, but we wanted to know what about migration, because migration is also pathological phenotype, and there is also often a link between proliferation and migration. So the way we assess migration was doing a scratch assay. So just doing a scratch in the culture and seeing if the cells migrate towards the middle to close the scratch. So what we saw here is that it's 5 out of 7 of the microRNA mimic show a significant decrease of migration, but you can see that the other 2 are actually close to significance. Overall, we think that at least for most of them, there was also an effect on migration, which might be quite interesting to use as well and to revert the pathological phenotype. And as I mentioned, it was also important to show that there was no toxic effect. So we use different measurement of toxicity. So the first one is cytotoxicity using a lactase dehydrogenase assay. Again, when we use the miRNA mimic, there's no significant increase of toxicity. On the right, it's our positive control. We look at senescence, again, no significant changes of senescence, and same for caspase-3 as a measure of apoptosis. So overall, we clearly see a change in proliferation, migration, but there is no toxic effect of this miRNA mimic. So all the experiment I show you has been done with 50 nanomolar of mimic. So the reason we kept 50 nanomolar is because the screen was designed with this concentration. I think now some of the screen probably use a lower dose, maybe up to 25, but 50 is still standard. However, when we went into the revision process, a lot of the reviewer commented like 50 nanomolar used quite in the high range of concentration. Say, are you sure you don't have any cytotoxicity? Are you sure the effect is really specific? So we decided to reduce the dose and assess proliferation. And as you can see, this is for miR-449B. Using a 2 nanomolar concentration, we still have a significant increase of the miRNA. And if we look at all of the miRNA mimic, even at 2 nanomolar, all of them show a decrease of proliferation. It's measured by the level of MKR67 by RT-qPCR. So it's an interesting result that came from the revision. I wish we had started with this lower dose because now most of the results are 50 nanomolar, but it means that we probably, if we want to develop a therapy, think about the dosage and that maybe scope to lower the dose later on. So this was all in terms of phenotype, but we wanted to know what was happening in terms of transcriptome. So we perform RNA sequencing. So the first thing we look at is principal component analysis, just looking at how similar the different samples are to each other in terms of transcriptome. So we sequence quiescent cells here on the left, and you can see that all the cells that have been treated with IL-1 and PDGF-beta are all on the right. So it means they are really different from the quiescent cell. If we look closely, we can see that all the controls, so IL-1 alpha, MOG, or miR control at the bottom, and all the cells transfected with the miRNA mimic are at the top. So basically, the miRNA mimic are changing the transcriptome, is distinct from the control, but they're not reverting to quiescence. What we know, they're stopping proliferation, but doesn't mean we end up with quiescent cells with the treatment. So we look at in more detail. So for every miRNA, we did a differential gene expression analysis. So we compare the cells treated with miR323P, for example, versus miR control. So this is a volcano plot, show you the genes that are up and down regulated. And we pointed here a lot of cell cycle genes. So as expected, we have a change of proliferation. So we expect cell cycle genes to go down. So this is what we see. And when we look at the enriched GO-terms, they were all related to cell cycle, which I suppose it's something we expected, but it was nice to see them at the top. And there was really nothing else except the cell cycle. And we could see that for all of the other miRNA. So we ask, okay, what is the reason we see exactly the same GO-term? So we compare the list of downregulated and upregulated genes. So there's only 3 common upregulated genes. But in contrast, there is 125 common downregulated genes by the 7 miRNA. And actually, when we look at this 125, there are a lot of cell cycle genes. So this is here on the right, the KEG pathway, the cell cycle KEG pathway, and all the ones that are in red are actually downregulated by the 7 mimic. So it shows that for all conditions, there is a common cell cycle network of genes that is downregulated. But of course, we know that they have this proliferative phenotype. Then the question is, how does this miRNA achieve this downregulation? Is it like a direct or indirect mechanism? We know how miRNA work, so we wanted to see, can we identify their target? So our strategy was to look at genes that were downregulated. So if the miRNA is overexpressed, they're going to bind to their target and trigger their downregulation. And then we use prediction tools. So prediction tools predict the binding site of the miRNA to the 3' UTR of their target. because some of the prediction tool, because prediction tools in general are not very reliable, we decided to test, to take targets that were predicted in at least prediction tools, so in the top 50% of at least prediction tools. And the overlap here are actually our miRNA candidate targets in vascular smooth muscle cells. So the table summarizes the candidate targets. They're quite viable. So miR5681 has got only 13 targets, while miR1827 124. And then we ask the question, are the targets similar or different between the different miRNA? So here's the heat map. You have on the left all the condition, and at the bottom, you have the target for the different miRNA. And the way I've designed this heat map is to have the ones that are unique for each miRNA on the left and the ones that are shared on the right. So what you can see clearly is that the shared targets are really a small number. Only 10% of the total number of targets are shared by at least 2 miRNA. And actually, there is no common target for all 7 miRNA. The only gene we found regulated by 5 miRNA was the gene IGF-2BP3. But on the left, around 90% of the genes are unique to each miRNA. So you can see this group of genes here, they're only downregulated, so here in blue for miR323, but the expression doesn't change for the other miRNA. So this suggests that even if they regulate the same core cell cycle network, they do it through different mechanisms of action. So that was quite interesting to see. And this is how we submitted the paper, but then the reviewer came back saying, hey, can you tell us a bit more about what is the mechanism of action? You mentioned IGF-2BP3. Is it the one that's driving the proliferation? So for the revision, we decided to see if we can find some targets that drive the anti-proliferative phenotype. So ideally, we really wanted to have a lot of targets, but then it's a lot of work. So we decided to do a selection of 3 targets. So we selected IGF-2BP3 because it's a target of 5 miRNA, and it was already mentioned in the first submission, and the reviewer picked up on it. But then we took also candidates that were more specific to some miRNA, but also relevant to all smooth muscle proliferation of cell cycle, because we thought it would give us an indication that they're more chance to drive the phenotype. So we validated expression. This is an example for CCND1. So it's downregulated by 449B. It's not significant for 491, but I think it's just a question of power here. And what we did for all these targets is doing a knockdown using siRNA. So we're mimicking what's happening to them when the miRNA are overexpressed, and check if there's an effect on proliferation by measuring the percentage of EDU cell. And what was interesting is that only CCND1 could reduce proliferation. So it means CCND1 for miR449B could be one of the major genes that trigger or lead to a decrease of smooth cell proliferation. But in contrast, the gene that's targeted by 5 miRNA, it doesn't seem to have an effect. So it means that it's going to be miRNA specific, but we also think that it's probably the combination of multiple targets that drives the phenotype, which makes it quite difficult to study as well. But I think that was quite an interesting thing to study, and at least to have one candidate, CCND1, for some of the miRNA. So what we've done was in vitro in smooth muscle cells, but of course, if we use this mimic in tissue, is it going to have an effect on the endothelium? We don't want to stop proliferation or do any toxic effect to endothelial cells. So we used human surface vein endothelial cells and perform the same assay we did previously, look at proliferation or look at toxicity. But you can see that this time, if we use the miRNA mimic, only 2 of them significantly reduce proliferation. And again, the reduction is not as strong as what we see in smooth muscle cells. So the miRNA seems to have a specific effect on smooth muscle cell and not on the cilia cell. And we couldn't see any toxic effect as well. To confirm that, we performed RNA sequencing. So same way we did for smooth muscle cell, we did it in endothelial cells. And here the number of genes regulated were really low. So it means they don't trigger a big change to the transcriptome. So these are the number, and the gray hash area are the ones that are regulated both in smooth muscle cell and endothelial cell. And we also look in more details of the gene regulated in smooth muscle cells, so the 125 common, they change in endothelial cell, and you can see that there is clearly no pattern. So basically, they don't change in endothelial cell. So that was quite interesting. And of course, it still raised a lot of questions why this miRNA affects smooth muscle cell and not endothelial cell. So it's something that's been discussed in the literature, could be due to the difference of transcriptome, and just something that might hint why we have this difference. So I mentioned this target CCND1. It's a target and drives proliferation in smooth muscle cell, but the level of the target is not the same in smooth muscle cell than in endothelial cell. Endothelial cell is lower. So we suggested in the paper that maybe the difference of expression of certain genes like CCND1 will make a difference and explains the difference of phenotype we see between smooth muscle cell and endothelial cell. And of course, one of the big questions is, it's all in vitro, what's happening in vivo or in tissue? And this is not something we addressed during the first submission. So of course, the first question, the reviewer is like, what about in vivo? The only thing is, what we have as a model for in vivo is mouse. Pig is also a really good model for vein graft failure. However, only 2 of the miRNA, 2 of the 7, to complete sequence conservation in pig and mouse. So while we could look at these 2, if we wanted to look at the 7 candidates, looking at animal model was not possible. So we thought, let's see what we can do in human. So in human, there is an ex vivo model of vein remodeling. If we take the vein and pin it down, it will mimic the remodeling with a change in smooth cell proliferation. So we use this model, cut the vein in different segments, transfected the mimic, and also added EDU to look at proliferation. And after 7 days, we did some staining for TAPI, looking at smooth muscle with the marker MAH11 and EDU. And this is how it looked like. So in our miRNA control, so it would be like normal vein, you can see that there is a lot of EDU cells. They align quite closely with smooth muscle cell. But if we transfect the vein now with miR323 there is a strong decrease of proliferation. So the mark of smooth muscle cell is just a way to guide us to the layer of smooth muscle cells, so it's not directly colocalization. And what we did that for 3 candidates only, and we could see that 2 out of 3 show a reduced medial layer proliferation. So it was really interesting to see that at least in tissue, 2 out of 3 miRNA could affect proliferation. So there's a graphical abstract of the paper, just to give you a summary. So as I say, we focus on vein graft failure, but we think that some of our candidates can be relevant in other disease. It was quite important to start with an unbiased screen, so it means we had the best candidate. We confirmed they had no toxic effect in smooth muscle cell, but also kept the cilia cell layer healthy and happy. They block proliferation and also affect migration. And what we look at is a bit of mechanism of action. So we believe that miRNA have different targets, but it converges to change to the cell cycle gene network, even if they do it for different mechanisms. Right. So to finish, I just would like to give you an overview about, I already mentioned across the presentation what was part of the revision and what was not. So it helped it give you an idea about how it happened and how we end up with this full story. So it took 9 months to do the full revision. I think it's probably, we could have done it in a shorter time. Just as I mentioned, the people who were involved, it was often their side project. So trying to have that was like a combined effort. The main point we got from several reviewers was looking at the mechanism of action and the in vivo study. That was really two of the strong main points. We had some comments related to some misunderstanding on the screening strategy of the miRNA mode of action. So it felt maybe like a rush review where the question was like, didn't apply to the miRNA mode of action. But I think it was still quite important for us to clarify the manuscript, and maybe it was important to actually in the right section to re-explain how miRNA functions so everyone could understand. There was a lot of question related to the mimic and anti-mir. So one of the reviewers was asking to use anti-mir because, as you can see, we use mimic all the time. But to use anti-mir, we need the miRNA to be expressed in the first place. And actually, the miRNA we study, most of them don't have endogenous expression. So here we're looking at ectopic expression or exogenous expression of the miRNA. And if we really wanted to use anti-mir, we should have designed a screen with anti-mir in the first place. And that's really what we tried to emphasize. Our screen was really designed to keep the mimic and take them out up to therapy. Of course, there's always some question that's a bit, I don't know if it is out of scope, but a bit tangent. So we look at endothelial cells, and one of the reviewers wanted to see the effect by paracrine effect. So I think it's an interesting question, but I think it would require a lot more study. So we just did a simple experiment with conditioned media that we included in supplementary figure, but it's something we could pursue in the future. So definitely all the revision improved the manuscript. And I think it helps just to clarify, and also adding, I think the mechanism of action and the ex vivo was really important. So for us as a future work, we really want now to test all the mimic in the ex vivo model. We only tested 3, so we want to test them all. We've not done a 30-minute delivery, so it's something we need to try, is if we do only a 30 minutes transfection or just bathing the vein, is it enough to make a change of phenotype? And we also need to check if there is any toxicity. We could go towards an animal model for the concept miRNA. But again, if we want to keep these 7 candidates, we need to think a bit more about how to test them. Now we're using the miRNA one by one, but maybe looking at combination might be quite important to see if there is a better effect. I don't think it's necessary in vitro, but might be the case in tissue. And of course, there is a choice of delivery. So at the moment, we're using mimic. It seems to be fine. It's not affecting endothelial cells. But if we want something more targeted, maybe viral delivery, where we control the expression of the delivery of a specific cell type, might be better for in the long term. But it's something we're still, so we probably need to go towards regulatory bodies to have also their view about local delivery and in terms of safety as well. So thank you very much. And I put again the slide where I can show all the authors that contributed to this work.
Thank you so much, Julie. Thank you so much for the presentation. Let me see. We have a few comments already. Okay, I can take on the first question. Actually, if Miron, if you want to come in and ask your question yourself, otherwise I'm happy to read it from the chat. Okay, I think, let me read it. Maybe he has issues with his microphone. So fantastic work.
In the bus.
Pardon?
I think Miron says that he's in the bus, so maybe it's better to read.
Okay, so now don't worry, let me read the one. Fantastic work. Since the screen has a comment hiding this, therapy, you probably need to consider systemic effect. Any ideas about off-target effects these miRNAs might have? Are you planning to design specific delivery system to target specific cells? You kind of touched upon on this, but if you want to add a comment.
So, but I think so for off-target effects with the RNA sequencing, we have an idea, we have an overall view about what's changing. If there is some change that, for some of them, we can probably try to exclude them. But again, there is a question of concentration as well. So that's something we need to keep in mind. We're only looking at same time at the RNA sequencing, which would be quite interesting to see at the protein level as well, because we know that microRNAs don't work on the RNA level. For specific delivery to target specific cell, we are considering this option. At the same time, at least in vitro, there is no reason to believe it's going to affect endothelial cells, which would mean that might be quite safe to try in tissue and see that that's still the case. Maybe using a mimic that will be going to all cells might still be a good possibility, but because they seem to have an effect only on smooth cell. But we're also looking at just viral delivery, and with viral delivery, we might have more way to look at specific cell. But even for viral delivery, we don't have an easy way to target specifically some maybe some subtype of smooth cell, for example. So we have option here, but I don't think there is the best system yet.
No, definitely. So I think we had a question also from Noemi. Again, if you want to come in and ask your question, but if you are on the bus as well, I can read it. No worries, I can take the one on.
Sorry.
Thank you.
Sorry, I was trying to find a mute.
Oh, no worries, go in.
No, I'm just curious because did you? Because you were focusing in vascular smooth muscle cell, also some kind in endothelial cell. Well, what about the macrophages and also the differentiation of vascular smooth muscle cells to macrophages? So, do you think that it's important to take into consideration the role of macrophages maybe there, or what do you think about that?
Yeah, we didn't look at it, and I think that's an important point. So I think the model we're using in vitro is IL-1 alpha and PDGF. So it's not doing the transdifferentiation. It's mostly a model that is proliferation and migration. I think the transdifferentiation to macrophage has been well described for atherosclerosis. I'm not sure for vein graft failure how relevant it is, but I think it would be quite interesting to have a better look if we put this microRNA into cells that we push them to transdifferentiate, what's happening. We've not done that, but I think that would be an interesting part.
And then maybe I also miss it. Maybe that did you try to do the experiments with the inhibition? So not overexpressing these microRNAs, but the other way around, inhibiting them and see if you have more proliferation, more,
So we cannot do that because they're not androgeneously expressed. So the reason we have an effect on proliferation is because we apply the microRNA mimic, but the cell don't express this microRNA normally, so we cannot try to downregulate. So I think the only way we would need to do that would be maybe to change cells, and in the cell that they androgeneously express, if we do anti-mir, can we now? But that would be, I don't know if that would be answering our question. I think in the context of let's work on therapy. So it doesn't matter about so much that we are doing in an androgenous situation. What we want to have is a compound that have an effect on the phenotype, no matter if it's expressed or not. But we had this question a lot, and I think if they were expressed, we would have done it. But the candidate we picked don't have a strong expression, or at least or no expression in smooth muscle cell.
Yeah. Thank you for the clarification. Thanks a lot. Nice work.
And building on that, as you mentioned at the beginning, it's very interesting that the starting point was a therapy perspective. So you guys were interested into using microRNAs as a therapy. And as you mentioned, in the end, all of the top candidates were actually microRNAs that were not there in the test in the first place. So we're endogenous. And my question would be what do you, about how, considering how the field is moving, what you think we're going in terms of delivery options when it comes to delivering microRNAs? Because clearly endogenous exogenous microRNAs can be used.
Yeah.
how far will you think like from actually being able like to deliver them in a more like precise and targeted way?
I think it's quite difficult because it depends on the disease and the system. I think in our case, we're really lucky to have this local delivery. So we're probably also, we don't have to think too much about systemic effect. But at the moment, some people use a lot of mimic. So they still believe that probably even the delivery is not specific, the effect is specific.
Definitely. Yeah.
So I think that's the question. I think it's miRNA specific and disease specific. So I don't know if that's people are looking at different angles. There will be some advantage to a viral delivery. At the same time, mimic, we can put them on directly. So it's a bit easier, quicker to test. So I don't know. I think we're exploring at the moment the different options, but I think in our case, I still think the mimic can be a good option because of this local delivery and probably the fact that we're not gonna touch so many cell types, but in another context.
I mean, it's easy. Yeah, I see your point. Like in this context, since there is an ex vivo model,
Yeah,
there's just so many options. There's just so
exactly.
many alternatives. So it's like kind of easy also to rule out.
Yeah, if there is any toxic effect in
in
the
other
other
cell
cell
types,
types, we
yeah.
can probably check them a bit more easily if you compared to if you were injecting a miRNA mimic that might go to different organs. Here, we're opting that with this local delivery, they probably won't
Yes.
go to other places in the body.
So you nicely explained at the beginning as well, like the way vein graft works from a surgery standpoint, like the real life setup is the vein is explanted from the patient and sits for half an hour to 1 hour, like on the side of the bed while the operation is ongoing. And that will be like the window to actually target it with the miRNA approach in this case.
Yeah.
So actually it's not that ex vivo setup of the experiments is actually basically what happens in the real life surgery theater. So we're kind of close to what is the real setup of the operation going on. So yeah, that makes sense. If there are other questions, please feel free to write it in the chat or to come in and ask it.
I think there was a question from Fabio.
Yeah, Fabio, do you want to ask?
Yeah.
Let's see, Fabio is in. If not, let me start reading. He can comment later. If I remember well, cyclin D1 is very important for hematopoiesis. Do you think this may be an important element moving to in vivo models?
Good question. I was not aware of that. I think, yeah, we'll need to think a bit more about it. I mean, cyclin D1 is one of the targets for one of the miRNA. So I think we've not fully decided about which, at the moment, 7 candidates. We'll probably need to narrow down, then we're not going to take forward all 7 of them. So I think we're going to have to probably look at smaller group of candidates, look again at the RNA sequencing, making sure we've not missed anything, and maybe review some of the target and make sure that in other system. But yeah, I think that's quite interesting to know. So something to keep in mind.
Thank you for that question. So one final curiosity from my end, and then we can see if there are other questions. So you were mentioning, of course, now it's very interesting to see this network of 7 miRNAs acting on different pathways and blocking proliferation. If you had to narrow down the cocktail, let's say, from a bioinformatic point of view, I mean, how would you, I mean, which do you think would be like a good strategy to narrow down to 2 or 3 candidates, let's say, which is like the best combination or how to obtain like one good combination that is giving you the highest coverage in terms of anti-proliferative capacity?
Yeah, I think the way I would look at it is that probably if we find some that target both targets cell cycle genes already, there's probably no need to put them together because we already know one is enough to assess. So I think we also look at GOTEM. So for example, some of them are there's more genes related to migration. So for me, I would combine probably the ones that have a strong proliferative or cell cycle target, then combining with some that I could see more migration genes, some maybe the transdifferentiation one we had about bone. So I think that would be more looking into the different pathway and making sure they don't overlap too much, because if they overlap too much, you can think that actually it's redundant. There's no need to have them together. At the moment, there's no need to think about the combination in vitro. They all decrease proliferation of 95%. So we cannot go more than that. We're going to kill the cells. So I think in this case, I don't know if my idea would be probably to all do another screen and target maybe miRNAs that more important for migration or miRNAs that are more important for transdifferentiation. I think that would be a safer or better combination instead of adding more miRNAs that all target proliferation, because overall we probably want to target all the different aspects of the pathological phenotype and not just proliferation.
So you will advise like using different model systems and screenings to target different pathological phenotypes, and then taking the lead compounds, let's say the lead target hits, and mix them.
Yeah, I think that's probably the way I would go. Again, if we had find that each individually decreased proliferation of 50%, then that would make sense to combine them. But if they're already quite strong one by one, I wouldn't think that is the need to combine them, at least at this stage.
Yeah. Thanks a lot. So I think that Miro jumped off the bus just to ask the final question of the meeting.
Thank you so much. Sorry, we are moving back to Serbia. This is my last week in Luxembourg, and we are trying to arrange so many different administrative stuff. So I'm, all over the place. Anyway, thank you so much for this very, very excellent presentation. And I was just wondering if you managed to decipher which particular protein or gene is responsible for these effects. Would it make more sense just to target this one gene, or you think that it's not realistic that it's just one gene, and then really using microRNA because they tend to have a little bit broader effects in terms of several pathways at the same time? You partially answered my question just a few seconds ago, but I just wanted to kind of ask, does it make sense to maybe, because I think it's from the perspective of developing therapeutics, it might be easier to target one particular target in respect to possible adverse effects in the future and so on, or with systemic effects of microRNAs. So in that sense, would be more convenient to just target one gene?
Yeah. So we don't know, and it's quite difficult. So for example, we find CCND1 that seems to drive a lot of the proliferation phenotype. So if you knock down CCND1, there is a strong proliferation phenotype. However, when we look at the miRNA effect, they downregulate their target, but they're not doing the same effect as the siRNA. They don't regulate fully to 90%. So I believe that actually the phenotype is probably a combination of the downregulation of maybe a twofold change of several targets, and all together that leads to the phenotype. So maybe for some microRNA, we can point out one gene that leads, or that is the main driver of the phenotype, but I believe it's probably the combination for most of them. Then in this case, you would argue that it's better to have a microRNA instead of looking at targets. The way to answer that would be with something we thought about, is taking most of the targets and doing a secondary screen with siRNA for all the targets and seeing which one actually contribute to the phenotype. But again, if we do a secondary screen, it's going to be a siRNA, a strong downregulation of the genes, which doesn't really reflect what the mimic is doing. But I think that will still answer a lot of questions, like what among the target, how many of them contribute to the phenotype, and then deciding if it's better to focus on the target or the microRNA. So we don't have the answer, but I think it's probably a combination of both mechanism of action.
I just want to say thank you so much, Julie, for this wonderful presentation. I have a project in our lab that is very similar, and that is also cooking. So it was really nice to hear your presentation and see all of the stages in your study design, and also the comments from the reviewers. So, but I'm not looking at microRNAs. I'm just looking at small inhibitors of intimal hyperplasia. But when you're talking about in vivo models, so you said that you would like to take forward maybe one of the microRNAs that would be your favorite into in vivo models. What models are you thinking?
So there is mouse model of vein graft failure. So it's slightly different. The vena cava taken. The only problem is that we know that in this model there is a lot of inflammation, and a lot of cell death, and it doesn't really mimic what's happening in human. So there is quite a lot of limitation of the mouse model. So mouse model would be the easiest to go for it. What people use a lot is a pig model. So we're thinking of a pig model. It's just really expensive. So I think it's just going to be, if we want to go this way, we need to secure funding. But I think if I had to choose, I think we would move towards a pig model. But again, we could do that only for 2 out of the 7 miRNAs, because the other one are not conserved. But if I had to go to a model, it would be a pig model, which is quite similar to what's been observed in human.
Exactly, nice. And there's also maybe the athro component that you have to think about, right?
Mm.
Because then when you're moving in vivo, then you might want to maybe look at, let's say, intimal hyperplasia separately, which is just a normal proliferation. But if you want to maybe make it more realistically applying to patients, you also have to think of the athro that maybe is something on top, and you want to see how your microRNA is then dealing with that, I guess. What do you think?
Yeah, I don't know. I think it's going to be probably at a different stage, and I can't remember in the pig what would be the timing to look at, because that's also the question that there will come later. And I mean, we're thinking a lot about this question for going towards the clinical trial, but I think the whole issue as well with this therapy is the longer. If you look at really long-term time points, then it's quite difficult to secure funding for clinical trial, because you don't want 5 years before having an outcome. So I think it's something, yeah, we've not looked at. Probably pig would be a good model to look at, but I can't remember exactly what would be the good timing to look at that.
Definitely. I'm afraid like even the shorter time points will be like a lot of time, like and a lot of funding to be secure in order to complete that. So I think we have maybe time for a final question. I think one came in the chat. Nemanja, do you want to ask your question, or do you want me to go ahead? Let's see if they are unmuting themselves. So let me let me read it for you. No worries. Is the R code used in this study available online by any chance? I read your paper. I know that you mentioned the packages used in the data is available on CBI. So I was just wondering if there are resources that go into the code a bit more.
We've not put the R code online, but I'm happy to share. Yeah, if someone is interested into the detail, I can share scripts, no problem. We didn't put it. It's something we should do actually for some of the bioinformatic, but happy to be contacted and share the code.
Thanks a lot for that as well. So I think that would be, you guys can stay in touch to have those codes up and running. And I think for everyone else, thank you for joining today. It was nice to have you, Julie, today here, and also nice to have someone from the bioinformatics side from once. Thanks everyone for joining. Next Journal Club will be mid-March, and we will be giving you more details soon, so you know what's the next paper to be discussed. Thank you, everyone. See you next time.
Thank you.
Bye.